Welcome to the today's session. It's about the mixing method of biofertilizer with the carrier. First, we prepare a special media for a bacterial inoculants. In a regular amount, you can prepare a special media for the preparation of a starter culture. Because the first culture it is called starter 1. This culture is again inoculated into a large fermenter or a tank and then incubated at 30 degrees Celsius for 5 to 7 days by providing aeration as well as stirring. After incubation, the cells are harvested and check the bacterial cells count by serial dilution technique. And this technique can enumerate the number of organisms almost present in the fermenter. After that, the cell population it should reaches the amount of 10 power 9 cells per ml. At that time, this point is essential to check the bacterial count. After that, the bacterial culture is mixed with the already sterilized carrier material having the moisture of 35 to 40 percentage. In this process, it can be done by either manually or by mechanically. Suppose if it is a manual method, you may wear a sterile gloves for mixing this carrier with the bacterial culture. Otherwise, it may be mechanical one. It is that's no problem for mechanical mixing. There is no bacterial contamination may invade into the mechanical process. Second slide is about mixing of inoculants with the carrier material. Almost there are 4 types of mixing of inoculants methods with the carrier. One is a carrier based inoculant. Here the carrier is the cheapest material compared to other ingredients. So the carrier it may be ash or it may be peat soil or lignite or some other materials derived from the agro waste products. And the material is mixed with the inoculant is the basis for the everything. That's why carrier based method is the cheapest one compared to the remaining methods. And the second one is called agar based inoculants. Here the agar is a chemical compound. It may be combined with the inoculants. It may cause some defects as well as some modifications in the biofertilizers. That's why we prefer only carrier based inoculants. And third method is about the broth culture method. This is the easiest one as well as it may be contaminated because the broth culture it may invade the foreign material into the starter culture. That's why we prefer only a carrier based inoculants. The fourth one is about the dried culture method. Dried culture method is the costliest one because the another name for the dried culture method is lyophilized culture. We must maintain the culture in a refrigerated condition. New developments in biofertilizer productions are available. First one is a freeze-dried inoculant preparation in India. The freeze-dried method is called a lyophilized culture preparation. Recent days in India we have following the freeze-dried method of inoculants preparation. And second is a rhizobium paste preparation. This is available in USA. Rhizobium is a root nodule forming bacteria. It may be combined with some other chemical to form a paste-like structure. Almost is an easiest way to maintain in soil. So that's why in USA rhizobium paste culture is prepared. And the granular inoculant preparation is the third one. In here the inoculants are modified in the form of granular. That's why the granules like structures may implant with the soil to fix the nitrogen as well as phosphorus into the soil. And the fourth method is about pelleting method. Pelleting method is almost familiar in nitrogen as well as in USA. The fifth method is about polyacrylamide entrapped rhizobia formation in an acroscate as well as the pre-coated seeds are also available in the New Zealand. These are all the new developments as well as the forthcoming generation. We may promise for the inoculation success in a tropical legumes. Third slide is about the application methods. In this slide, we concentrate the application methods of soil. And basically, the most common way of inoculation is called a seed inoculation method. In this method, the inoculant is mixed with the water to make a slurry form and then mixed with the seeds. And second one, the carrier it must be a form of fine powder to bind or make thick coating on a seed surface. So the third case is about for this coating purpose, the adhesive material of gum arabic or methyl ethyl cellulose or sucrose solutions, vegetable soils are used. This method may not always be successful for small size nodule occupancy, and the strain have a low affinity with the adhesive materials. That's why it's not an effective one for the low nodule occupancy strains. This seed inoculation method almost enhances the chance of soil fertility and to prevent the predators or the nematodes to make a contact with the plant roots. And a second method is about soil inoculation step. For this soil inoculation, a granular inoculants are placed into the soil along with the seeds. Granular inoculants, already we familiar about the granular inoculants. So these type of granular inoculants are placed into the soil along with the seeds. That's why the carrier material with the granular form, it's almost 0.5 to 1.5 mm is generally used. The granular form of peat as well as perlite charcoal or the soil aggregates are suitable. This method almost enhances the chance of inoculated strains to contact with the plant roots. So the first method is to prevent the predator contact. The second method almost enhances the inoculated strain to contact with the plant root. This is an efficient one. That's why soil inoculation is the best one, and the seed inoculation to prevent the microbial predators growth. That's why both are beneficial as well as best methods. Next slide is about the overall production of biofertilizer as well as the quality checking method in this production process. So the many farmers in India are almost shifting towards the organic farming due to a domestic as well as the international demand for the organic food. The quality assessment of the inoculants have been examining the moisture content, viable count of organisms and the contaminants present in the biofertilizer and the effectiveness using a plate count as well as the MPN tests. First, the preparation of biofertilizer is always start with the inoculant or the starter culture preparation. First of all, an efficient nitrogen fixing strain is required, and this is inoculated with a small flask containing a suitable medium for inoculant production. Second, the media and the growth conditions like temperature, pH and the time duration for the bacteria, it must be optimal, and also ensure that the cells are in good physiological condition. That's why it's termed as starter culture for everything. A starter culture or a mother culture should be suspended in a liquid as well as freshly prepared medium. The volume requirements of the mother culture in a test tube, it must be in 1:10 ratio. For example, if you are inoculating a 1 ml of culture into the 10 ml of liquid broth, this is called 1:10 ratio. This may be done in a liquid or on a solid media by multiple stage process. If a large fermenter is used, you may use this multi-stage process. At present, the biofertilizer is supplied as a carrier-based microbial inoculants, which are added to the soil and enrich the soil fertility. Here, the carrier must be in a sterile condition. Other important step in a quality assurance is biofertilizer formulation. Now, the new carrier materials are needed to overcome these limitations, like availability, environmental impact, toxicity, and provide a suitable environment for the microorganism. These are all the new challenges we are facing in the biofertilizer manufacturing. And the carrier is a medium that can carry microorganisms in a sufficient quantities. Keep them available as well as viable in a specified conditions, as well as packaging and sealing follow some rules and regulations. Finally, we may supply this biofertilizer to the farmers. Before and after delivery, the quality of the biofertilizer should be checked and stored at low temperature until it reaches the farmers. In a previous one, we discussed the quality checking method by plate count or by serial dilution. we have to follow the other major aspects of quality assurance by maintaining the cell viability during the transport as well as in a storage mode of application. The mode of application like a seed coating, on-site seed application or soil inoculation is the best method. Almost all the biofertilizer companies manufacturers follow these methods. This may also affect inoculation efficiency depend on kind, variety and the size of the crop as well as the fragility of the seeds.